Today we have Assistant Professor Marc Fivaz from the Laboratory of Neuronal Morphogenesis and Plasticity. Marc's lab is part of the Neuroscience and Behavioural Disorders program, which is one of the 5 signature research programs here at Duke-NUS.
So the main research focus in the lab is to understand how neurons acquire the incredible complex and beautiful shape during brain development. And we're also interested in how the shape of neuron is modified by experience or learning or altered in neurological diseases, such as, for example, schizophrenia or Parkinson's disease. Now, this is an important question for both basic and clinical research, and this is because the shape of neurons is directly linked to their function. For example, proper synaptic transmission depends on the ability of neurons to grow an axon and dendrites and to build a functional synapse. And there's also now increasing evidence that subtle alterations in the structure and the architecture of neurons are associated with neurological disorders. So it's a very important question. It's also a very difficult question to crack or to answer. We have access now to the full human genome sequence, but we're actually very far from understanding how the genomic code is actually translated into a program that builds a neuron or an entire brain for that matter. So the challenge now is really to understand how these genes and gene products interact in space and time to bring about such complex structural changes and how this is altered in diseases.
So Marc, what area of neuronal development is your lab focusing on?
We're focusing on 3 main areas, and the first topic deals with axon growth and regeneration. So as you know, adult neurons of the central nervous system don't regenerate, and this leads to permanent and devastating disabilities after spinal cord injuries or strokes or neurodegenerative diseases. Those very same neurons, when young, have a remarkable ability to grow an axon early on during development. And it's actually not known why a neuron loses its ability to grow an axon as it ages. So our approach to this problem is to try to identify a signaling network or signaling circuits that promote axon growth early on during development. And the long-term goal of this project is to reactivate these circuits in old neurons to promote regeneration after injury. The second topic deals with synapse formation and synaptic plasticity. Now, synapses are these tiny cellular junctions that allow neurons to communicate with each other. You have about 100 trillion synapses in your brain. And so it actually turns out you have more synapses than there are stars in our galaxy. So we're very interested in how these synapses are assembled during development, how they're remodeled by experience or by neuronal activity. And we're focusing in particular on the role of calcium signaling and microRNAs in this process. And finally, the third topic is about axonal transport. So axonal transport allows a neuron to deliver material from the cell body to the axonal growth cone or synapse, or back from the synapse to the cell body, sometimes over very long distances. Axonal transport is very important for a neuron to function properly. And it turns out that axonal transport is very often disrupted in a number of neurodegenerative diseases, such as, for example, Alzheimer's disease or Huntington's disease. Now, disruption of axonal transport is thought to occur very early on in these diseases and could potentially contribute to pathogenesis and disease progression. So what we're doing in the lab is we're trying to understand or study the type of axonal transport deficits that you have in mouse models of Parkinson's disease, for example. And we're also currently testing the idea that axonal transport deficits might also be associated with neuropsychiatric disorders, such as, for example, schizophrenia.
Give us the types of model systems and techniques that you use in your lab.
Sure. So we work with the mammalian central nervous system using neurons either from rats or mouse. And to study the impact of a single gene on neuronal development, we typically use RNAi interference silencing techniques. We also have access now to knockout or transgenic mouse models thanks to our collaborators. Now, one technique that we extensively use in the lab is live cell imaging. And the reason we use live cell imaging is that live cell imaging really enables you to watch a biological system function over time. And so it is a very powerful technique to study the impact of a gene deletion or a gene mutation on neuronal development and synaptic functions. Now, we also use and developed a series of fluorescent biosensors that we use to measure important neuronal parameters, such as, for example, synaptic activity, neurotransmitter release, or axonal transport. And in addition to that, we also use fluorescent biosensors to look at the activity or measure the activity of important signaling molecules inside cells.
How is your work relevant to neurological disorders and neurodegenerative diseases, and how will our research make a difference in this area?
So for most of those diseases, we are at a stage where we still need to understand the fundamental mechanism that contribute to pathogenesis. Now, for this reason, a large part of our work aims to understand the fundamental and basic mechanisms that govern neuronal development and synaptic functions. Now, having said that, we have a couple of projects that have more immediate relevance to clinical research, and I just want to tell you a little bit about two of them. So the first one is about axon regeneration. So we have identified a signaling circuit that promotes axon growth early on during development, and we're now in the process of testing whether reactivation of the signaling circuit in old neurons is sufficient to promote regeneration following injury. And in collaboration with Takanori Inoue at Johns Hopkins University, we're thinking about ways to reactivate this signaling circuit using a pulse of laser light. So another way to look at this is that we're trying to turn an old neuron into a young one using light. Now, the other topic is about axonal transport defects in neurodegenerative diseases. And for this work, we have developed an imaging platform that can score very accurately axonal transport deficits. And so with the recent development of stem cell technology, and in particular iPS cell technology, we are now in position to sort of monitor axonal transport defects in neurons that are directly derived from human patients suffering from those diseases. And so we think that this approach could be an important step forward in order to develop new diagnostic tools, but also new disease models. And so we're very open to collaborations in this area, and we're looking for groups who would be interested in monitoring axonal transport defects in their favorite disease model.
Give us an example of some of the collaborations that you are doing with other institutions, or you are about to be doing with other institutions.
Yeah, sure. So we have ongoing international collaborations with labs that are located in the US, in Japan, and also in Italy. And we also have, of course, collaborations with researchers here in Singapore, and these include groups at NNI, at NUS, or of course here at Duke-NUS as well.
How big is your research group?
So we have 3 postdocs, 1 PhD student, and 2 research assistants. And we're always welcoming new PhD students. And if you're interested in our work or in the work that we do in our program or at Duke-NUS, you should consider or think about joining the IBM PhD program.